morning, dear students. Today we are going to complete what we started in the last week. In the last week, we started in theme 1, concept 1, lesson 1, the cell as a system. We talked about the cell, the meaning of it, the components, and the function for the cell membrane only. Okay, and we said also that the living organisms, their bodies consist of many cells which are very tiny particles by using a special tool which is called the microscope. Today we are going to start in lesson 2, the compound microscope. Also, although we know that the cell is very tiny, there are other cells are very big. We can see it with our naked eye. But there is a scientist called Robert Hooke who invented the compound microscope, as we see in 17th century in 1665. And he called the tiny particles that the bodies of the living organism consisted of it with the cell. So, who is the scientist that discovered the microscope? I can write Robert Hooke in 17th century, or we can say in 1665. What about the importance for the microscope? If there is no microscope, can we examine the cell? No. Can we examine all the components of it? Also no. So if I want to see the details for any cell, actually for the plant cell, the animal, and also the human, I can use the microscope. There are different types of the microscope, like the compound one, like an electronic one, but when Robert Hooke started invention of the microscope, we started with the compound microscope. What about the structure of the compound before using it? I have to know what does the microscope consist of. It contains many parts, starting with the eyepiece, in which the part I can see the cell through it, coarse and fine focus, that we make the cell very clear, arm, we can hold the tool from it, stage, I can put the slide on the stage and fix it with the stage clips. The tube, also the objective lenses that has different sizes. One of them, or we can say two of them, are low and the other one are high. When I examine the cell, I starting the lower one, then the higher one. Like when you take a photo with your mobile, you can take a photo with a very small size, then you can maximize it to see its details, and the same for the microscope. Also, we have a mirror, because it is the first microscope that is invented, actually was invented in the 17th century. So starting with the mirror, or we can say illuminator, and the advanced one, we can work it with the electricity when I put the wire in the socket. Also, we have the base that carry all the parts of the microscope. This is the structure for the compound microscope. Okay, I have the microscope, but how can I prepare the slide to observe the components for the cell? For example, a plant cell, we can use the onion, the potato, any plant cell. I can use it for an example to examine the plant cell. I will use the forceps, the dropper to drop some water on the slide, the glass slide, of course, I have to use the glass slide to put the cell on it, and also the cover slip. As we see, I can put the cell on the glass slide and also cover it with the cover slip. Okay, how can I prepare the cell for examination? The first step, I have to cut, or we can say separate, a very thin cell membrane from the onion by using the forceps. Then I can put it on the slide and add a drop of water by using the dropper, and finally I will cover it by using the cover slip. Now I prepare the cell well. Now what should I do to use or to examine the cell by using the microscope? I can take the cell that I prepared before and put it on the slide, then fix it by using the stitch clip. Like the nail, if you want to fix any picture on the wall, you can use the hammer and the nail to fix it, right? Also the same for the microscope. I can put the slide on the stage and fix it by using the stitch clips. Then I can use the lower objective lens. Don't forget, I will use the lower first, then I can maximize it by the higher one. So I will put it and point the lower objective lens to the slide and make the image clear by using the coarse focus and also by using the fine focus. The coarse and the fine focus make the cell very clear. Maybe it will be not very clear well to see it, so I can use the coarse focus and fine focus to see the slide, and finally I can prepare between or use the lower and the higher objective lens. Then I can see the cell as we said from the beginning through the eyepiece. What will happen? What will we observe after putting the slide on the stage of the microscope? Number one, I can see the cells by using a lower objective lens appear in a smaller size. And the second step, by using the high power objective lens, the cell as we see appear in a bigger size. This is the difference between the low and the high. It gives us different degrees of the pictures. So today we learned how can we prepare the slide and how can we use the parts of the compound microscope to examine the components of the cell very well. Let's answer some questions about what we talked today. Let's see. The questions can come like this way. I will bring a picture for the microscope and you will label or write the name for each part. How comes? First, when I ask you this figure represents what? Excellent, very good. I will write the compound microscope. Okay. Letter A represents what? The eyepiece. I can see the cell through it. Letter B, objective lens. Letter C, stage. D, the mirror, or we can say illuminator. E, the base that carry the microscope, and also F, the arm that I can hold the microscope from it. Again, eyepiece, objective lens, and in letter B I will not write which one is lower and which one is higher, just write the objective lenses only. Letter C, the stage, but if you point in the clips I will write stage clip. But here it points to the stage only. The illuminator or the mirror, because it worked with the light, not with the electricity. And letter E, the base that carry all the parts of the microscope. And letter F, the arm. That's for question 1. Let's see another type of question. Choose the correct answer. 1. Was the scientist who used the word cell or who named the tiny particles by the cell? Excellent, of course, the Robert Hooke. There is no Robert here, so I will choose the Hooke. Okay, this is number 1. What about number 2? The modern microscope helps scientists to discover all the following information about the cell, except what? And at this question, I have to write all the choices well and choose the most accurate one. The cell is the building unit of the living organism. Yes, true. Some simple living organism consists of only one cell. Also right, like the bacteria. Letter C, living organisms that contain complex systems or many cells. Yes, like the animal, the plants, and also the humans. Letter D, all living organisms have the same parts which have the same function. Oh, here I have to think well. Do the living organisms consist of only one cell or many cells? Of course, many cells, trillions of cells. When we said before in the lesson 1, we said that the living organism consists of 40 trillion cells. Like, do the cells have the same function? Of course not. The function for the eye is completely different from the mouth, the ear, the nose. Each part or each organ has its own function. So, which sentence here is incorrect or is false? Excellent. Letter D, which is all living organisms have the same parts that have the same function. Of course, it's wrong because our bodies contain many cells and each cell has its own function. Okay, let's read letter 3. All the following are from the parts of the compound microscope, except what? Let's see the parts here. Eyepiece, it's also found in the microscope. Objective lens that I can use to change the degrees for the cell from smaller to higher one or bigger one. Illuminator, yes, we saw before that there is an illuminator inside the microscope. Objective mirrors, there is no objective mirrors here in the compound microscope, so all the following are found inside the compound microscope except the objective mirrors. You can change the power magnifying microscope by using what? If I want to play between the sizes of the cell by using different degrees, which parts of the microscope I can use it to change the size of the cells? Of course, the objective lenses. As we see, the objective lens. Objective lens here has a lower and also the higher. Maybe gives us a smaller size or a bigger size. This is a question that I can use it or you can see it if you want to answer some question about lesson 2. So today we talked about theme 1, concept 1, lesson 2 about the compound microscope, the scientist who invented it, who named the tiny particles with the cell, the structure of the compound microscope, the function for each part, how can we prepare the cell, how can we use the microscope, actually the compound microscope, to observe the cell, and also we answer some questions in the booklet about lesson 2. I want or I wish to understood well the lesson. Wish you a happy day and goodbye.